to be very well tolerated, and that enables us to combine it with such things as chemotherapy in our first-line colorectal cancer program. Now, you can see here on the slide as well that it is a small molecule, and very importantly, it's oral. And also on the right-hand side of the slide, we're showing that there are other PLKs that are evolutionarily related, PLK2 and 3, but if you see the 10 versus 0.002, that's the 10 divided by 0.002, that's a 5,000-fold greater specificity for PLK1 than other PLKs. And we believe that that's one of the central reasons why this drug, onvansertib, is so well tolerated and really separates it from past PLKs, which were pan inhibitors that hit all the PLKs. So with that, I think what we're going to do today is really three things. We're going to first talk to you about our first-line program, which Roger will be doing. Very exciting data. I will come back, if we have time, to talk a little bit about the incredible and exciting new mechanisms of action that we discovered along the way that got us to this first-line trial. And then Jamie will come in and really close the session talking about our coming catalysts and cash. With that, Roger.
Thank you, Mark, and also thank you to Noble for this opportunity. Here we wanted to provide just some background on the disease setting. We're talking about colorectal cancer, which is the third most common cancer worldwide, and unfortunately is actually increasing in incidence, especially in younger people under the age of 50 in the United States. In terms of when patients have metastatic colorectal cancer, the median progression-free survival, or PFS time, is usually less than 12 months with a relatively low 5-year overall survival time. In particular for RAS mutated MCRC, which represents approximately half of the overall opportunity in colorectal cancer, there has been a dearth of innovation for these patients with the only targeted medicine that's been approved in combination with chemotherapy, that is bevacizumab, which was 20 years ago. So we certainly have a very high unmet need situation here. And to talk a little bit about KRAS and RAS mutation, as some of you may know, there are specific inhibitors of specific mutations in KRAS that are approved in colorectal cancer in later lines of treatment, but target a very narrow subset of the overall population. In terms of the RAS mutant subtype, it's approximately half of the MCRC cases that are seen clinically, and onvansertib has the opportunity to address all of the known RAS mutant MCRC based on its mechanism of action. In terms of the clinical picture, we wanted to provide some background in terms of what have either molecules that have been approved or had positive phase 3 studies reported in terms of the objective response rates in particular that were associated with clinical benefit in the RAS mutant population. And what we learned is that these are actually have been associated with fairly modest improvements in objective response rates, but were strongly associated with improvements in progression-free survival. So just some numbers to keep in mind as we go through our clinical presentation of our data. So the Cardiff 004 trial is a phase 2 study in the first-line RAS mutated metastatic colorectal cancer. The primary objective of the study was to show proof of concept in combination with chemotherapy and bevacizumab, and to select a dose to move into a registration-enabling phase 3 studies. The enrollment criteria are listed: previously untreated metastatic colorectal cancer, no prior bevacizumab, and it's a 6-arm trial. 2 different chemotherapy backbones, of which are commonly used in the US, FOLFIRI bevacizumab and FOLFOX bevacizumab, and 2 different doses of onvansertib at 20 milligrams and 30 milligrams. The primary endpoint is objective response rate, with secondary objectives of duration of response and progression-free survival. And importantly, response is assessed by blinded independent central review, and patients' tumors are scanned every 8 weeks. So in terms of the key data that we updated in July, we actually are able to show the confirmed objective response rate of 49%, which is a nearly 20% improvement in over the control. And we think that that is certainly clinically meaningful, especially when you take into account the previously reported positive phase 3 studies in the first line RAS mutant population that we showed on a prior slide. This slide is actually also the waterfall plot, which shows patients' best response on trial. And you can see compared to the control for the 20 and the 30 milligram arms, we are certainly achieving significantly better depth of response that we know is correlated to improvements in durability like PFS down the road. Another great way of showing what is happening over time is the spider plot. And we can see in the control arms on the left that it is active chemotherapy and patients do respond. And you can see the change from baseline in terms of the tumor reduction on the y-axis over time on the x-axis that patients are responding, but tend not to deepen those responses over time. Whereas when we have the onvansertib added to standard of care, you can see a very impressive difference in terms of patients having complete responses, even 100% partial responses, and improving the depth of response over time beyond just the RECIST 30% requirement. And we've spoken to many different key opinion leaders in the metastatic colorectal cancer space, and they have been the most impressed by the depth of response that we're seeing. They don't commonly see this when adding agents to standard of care, and we know that it's strongly associated with improvements in durability with more follow-up. And just to give you a sense of who were our best responding patients, we wanted to know if, for example, these were maybe patients who were likely to respond to chemotherapy anyway, possibly with low burden disease. But this actually wasn't the case. We actually learned that when onvansertib was added either to FOLFOX Bev or FOLFIRI Bev, either at the 20 or 30 milligram doses, patients that would be considered difficult to treat had these incredibly deep responses. Patients with not just target disease in the liver, but with more advanced disease like disease in the peritoneum or even disease spread in distant organs, which we think really speaks to the power of the response that we're seeing. And some of these patients were actually referred to curative surgery when they originally were considered unresectable. We also reported the progression-free survival time, or PFS, and we considered this immature at the time based on the degree of follow-up. We had approximately a median follow-up time of 6 months for patients at this data cut. And we know from the disease history that we would expect the median progression-free survival time to be anywhere from 10 to 12 months in this disease. So we did encouragingly see some early signs of separation in the PFS curves, especially for the 30 milligram dose versus the control. But we expect to be more informative with more follow-up, in particular in the Q1 timeframe when we get closer to 1 year of follow-up on all the patients. What we can say is that, as I mentioned before, in the metastatic colorectal cancer literature, it's been published in some of the top journals, large retrospective analyses in terms of how both early tumor shrinkage, which is defined as at least a 20% reduction in tumor size at the first imaging time point, and also depth of response are strongly associated with improvements in progression-free survival. And these analyses were done retrospectively across both bevacizumab and EGFR studies that combined with the similar chemotherapies that we are studying in the 004 trial. Another important characteristic is safety, and we have, in a great way, we're actually seeing very little additive toxicity on top of standard of care chemotherapy. We would expect from the mechanism of action, one of the important adverse events of interest would be hematologic, in particular events of neutropenia, and we've highlighted those in the chart. And especially in terms of grade 3 or higher adverse events. We're not really adding much to the background chemotherapy rates as we observe them in the trial. And for example, with neutropenia, we haven't seen events of febrile neutropenia or discontinuation of onvansertib due to onvansertib related adverse events. And we think this is also important for durability down the road, as patient compliance with therapy has been relatively high, given how it's very well tolerated and easy to administer, given it's an oral medication on top of standard of care chemotherapy. In terms of next steps, as we highlighted, the 004 study really was imagined and designed after interaction with FDA in 2023, where a novel finding in the second-line metastatic colorectal cancer was observed, where patients that had not had prior bevacizumab seemed to have much better response. And that has led to the 004 study, and we also were able to discuss the key elements of a phase 3 program that employ an objective response rate for accelerated approval and progression-free survival for full approval. And we're in the process of designing a phase 3 program or a clinical trial that we plan to discuss with the FDA. And with that, I will turn it over to Mark to talk about the rationale for our shift from second line.
Well, thank you, Roger. And I think the question really comes down to, we have this exciting data that Roger just showed you, these really deep responses, 100% decrease in basically tumors going away. But what is the fundamental mechanism of how is this happening? And so this is actually another journey that we had, that we started obviously prior to this 004 first line data that you just heard. And it really started by looking at our understanding of what was the synergy between onvansertib and bevacizumab or Bev or Avastin. And you can see here, this is a very typical experiment done preclinically where you put human tumors on a mouse in the hind leg, and then you measure the growth of that tumor and versus what you give that mouse. And you can see here that on the curve here, on the y-axis, you're looking at tumor volume, and on the x-axis, looking at days. And you can see right away that onvansertib plus Bev gives you much greater control and an inhibition of tumor growth. So we just said, well, that's very interesting. Let's take a look at a couple more. And we kept seeing the same thing over and over again. That is to say, onvansertib and Bev were somehow working in concert to really inhibit tumor growth. And so we actually asked the CRO that was doing the study, we said, you know what, why don't you do gross dissection at the end of the study and take a photo of the actual tumors and send that to us. And they did. And what you can see on the top is a very large, bloody kind of tumor, kind of gross. But if your eyes go to the bottom, you will notice right away that the combination, not only are they a lot smaller, but importantly, they're really pale. And what that meant to us was there's something going on, that onvansertib was working in concert with Bev to be able to inhibit angiogenesis. That is to say, vasculature coming in and basically feeding the tumor. It was inhibiting that. So the next slide really shows you a lot of work that what we did. We then began to understand what was happening was that one of the very well-known hallmarks of cancer and tumor growth is that a tumor grows so fast. On the left-hand side there, you can see that it basically it becomes hypoxic. It doesn't have enough oxygen. And so what does it do? It survives, it adapts. How does it do that? By increasing this whole new genetic program that was really started by this HIF1 alpha transcriptional factor at the top right. That then drives all kinds of new genes to get turned on. One of them is VEGF-A, which, as we know, is inhibited, neutralized by Bev. VEGF is an angiogenic factor. It detracts vascularization. Well, what about onvansertib? Where does onvansertib fit in? Well, it actually is at the master switch. It actually shuts down the whole program. And so these two work in concert we believe, this is the reason you've seen the underlying clinical data. The next slide really shows then, this whole work was actually published last year in ASCO's flagship journal, JCO. And on top of that, because it was a novel finding, we actually have two new US patents issued that actually give us runway in colorectal cancer out to 2043. So this has really we've been very excited about this. And with that, I'm now going to turn it over to Jamie to talk about catalysts and cash.
Thanks, Mark. And again, thanks to Noble for allowing us to be here. Before I talk about cash, I thought I would just comment quickly on the relationship that Cardiff Oncology has with Pfizer. Back in 2021, Pfizer made a $15 million investment in the company. They currently hold a little over 3%. And importantly, along with that investment, Pfizer does appoint a member to our scientific advisory board, and they also have the right to see data before we make it public. 2 days before we make it public, we show the data to Pfizer, we get their feedback. When Pfizer saw in 2023 the fact that we were moving into this very large patient population of first-line MCRC, RAS-mutated MCRC, they proposed to us, what you see on the right side of this slide, that Pfizer Ignite act as the contract research organization, effectively acting as the entity that would run the trial. Now, we are paying Pfizer in cash like we would pay any other contractor that would assist us in running the trial, but it does show the strategic interest in this molecule that Pfizer has. And importantly, we still retain full ownership and control of onvansertib. Pfizer does not have any rights in that area, but we do have a close working relationship around the 004 trial. Finally, looking at our cash and investments as of the end of June 30th, 2025, we had $71 million in cash. We've said that that cash funds us into early 2027. Now, we've publicly guided that the next update from the 004 trial will be in the first quarter of 2026, so next quarter. And the cash that we have on hand funds us into early 2027. So we feel we are well funded with a year's worth of cash after the next significant catalyst to fund the company's operations and to make good decisions about the clinical path forward for onvansertib. So with that, I think we'll be turning it back to Robert, who can provide some Q&A.
Well, thank you very much. That was a very interesting presentation. And one of the first questions is that when you're adding this PLK1 inhibitor, the polo-like kinase inhibitor, you're adding another mechanism of action to the standard of care, the FOLFOX and FOLFIRI with bevacizumab combination. Given the wide range of cancers where onvansertib could be effective, how did you choose to focus on the RAS-mutated metastatic colon cancer?
Yeah, I'll take this one. This was a great question, Robert. You know, it really started at Harvard. One of the Professor Elledge at Harvard published over a decade ago that there was a unique interaction between PLK1 inhibition and basically tumor cells from the colon, colorectal cancer tumors, that had a RAS mutation. That is to say, if you had a RAS mutation and you also inhibited PLK1, those cells died much more readily than if it was a RAS wild type background. That really got us started. That and the fact that in colorectal cancer, we knew that irinotecan, which is FOLFIRI, one of the chemos, we knew that had basically was synergy, had synergy with onvansertib. So we really put our first bet really in colorectal cancer because of the basically the synthetic lethality between RAS mutation and PLK1 inhibition and the synergy with the chemotherapies already given in colorectal, and that's where we got started. And that's why our focus is really in this area. But like you asked, there are other places that we are also looking at really going after simply based on some of the mechanism we just talked to you about, and also synergies with other chemotherapies as well. We recently basically presented work in triple negative breast cancer at the most recent ASCO conference. Very exciting data in triple negative breast cancer. We also have single agent activity in small cell lung cancer. another very exciting area that we're looking at very closely. And we also are looking at other GI tumors. We're also looking at in essence, the other parts of colorectal cancer, like even wild type, because we think there's actually something going on there that's different that we just talked about as far as RAS mutant. So anyway, I think the answer to your question is that onvansertib is very well tolerated and is central to many different mechanisms that we can really leverage into going after and killing these tumor cells.
Okay, do you have any plans to start clinical trials or any efficacy data in these indications?
We do. We've already started investigator-initiated trials. That was what was reported on triple-negative breast cancer, work out of Dana-Farber at Harvard. And that was really showing the activity of onvansertib in combination with paclitaxel. Another very interesting basic science there of how that combination kills tumor cells. We reported that. And then we do have other like investigator-initiated trials in small cell lung cancer, as well as in pancreatic cancer. So we're doing those. We're watching our resources, and to be transparent, our focus is really on the colorectal cancer program in first line, since we're seeing such exciting data there. But we are also exploring some of these other areas as investigator-initiated trials.
Great. And you mentioned that there's an update from the trial expected in the first quarter of 2026. Can you tell us what to expect from that update?
Pass that over to Roger.
Sure. That's a great question. As we mentioned, there's high interest in not only the strong objective response rate data we're showing in terms of the improvement over control of nearly 20%, but also measures of durability, specifically duration of response and also progression-free survival. And I think that this update that we provided was in the July timeframe, approximately 6 months of follow-up. Towards the first quarter, we should get a lot closer to a year of follow-up, which we are hopeful will be more informative with respect to kind of the key measures of durability from the 004 study.
Okay, and we have a question from the floor, from the audience, asking whether all of the patients on the 30 milligram dose have completed their 6-month scans, and if there's any new data available for the cohort or any update on the ongoing RAS wild type.
So in terms of the patient disposition for 004, the study ended enrollment in April. And so we are expecting to have, as we mentioned in the Q1 timeframe, an update not only on the objective response data, but also on the duration of response and also for the PFS.
Great. Okay, and thanks for explaining the relationship that you have with Pfizer. Are there other companies that Pfizer's invested through the Breakthrough Growth Initiative?
Sure. So Pfizer did make several investments through the Breakthrough Growth Initiative. One of the more notable is Trillium, that was subsequently acquired by Pfizer. But when you look at most of the analyst reports, we certainly see this as a very significant drug and have dialogue with a number of the large pharma companies that are interested in GI oncology, which is particularly active in the pancreatic side. There's not as much available in MCRC, and obviously that's where we feel we have very encouraging data.
Yeah, and just from the uses of the drugs in the clinical trial and other regimens, yeah, I would expect quite a lot. But is there? Could you talk a little bit about the commercial opportunity and the patient population for metastatic colon cancer?
Sure. From a commercial opportunity, I guess I would highlight that most of the analysts that publish on onvansertib have a peak sales estimate somewhere between $2 billion and $3 billion a year. So a very significant drug. In our current clinical trial, we do not see competitive trials for our patient population. So we feel that in first-line RAS mutated MCRC, we have a pretty open field. Maybe the last comment I would make is, as Roger described, we're adding onvansertib to the current standard of care. We're not taking anything away, and we are showing that it's very well tolerated. So in the commercial consulting work that we're currently performing, it does, the projections that we have are that we could have very fast adoption of onvansertib, because if we show significantly improved efficacy, no additional toxicity for a patient population that has had nothing new in 20 years, we do feel like that sets up for a very attractive commercial opportunity potential for onvansertib.
Yes, I would agree with you. The fact that it fits in with their current practice for these patients and doesn't add additional toxicities is a key factor that I would expect the doctors would be looking for and affect the decision to add it. Are there other drugs in development that would compete with onvansertib?
So right now, we're not, as I said, we're not seeing competitive trials against our first-line RAS-mutated population. We do see that, as Roger mentioned, there are potentially drugs targeting G12C, but that's a small sliver of the RAS-mutated population. And right now, those aren't drugs targeted at the first line. They're in later lines of therapy. So we do feel like we have a fairly open road for the commercial development of our drug onvansertib.
Great. Okay. And just one other question about the market and acquisitions was that Merus announced that it was going to be acquired by Genmab for $8 billion. Does the Merus program in first-line metastatic colon cancer compete with your program?
No, thanks for pointing that out. And I know that in the past there has been a little confusion about this. As we said, RAS mutated is about half of the patients in first-line MCRC, and that's our target. Merus, in their CRC program, is targeting the other 48%. They're looking at RAS wild-type patients. So in that positioning, no, we don't see Merus as a competitor because they're only focused on the wild-type population.
Great. Okay, well, just in the interest of time, I'd like to thank you for participating in the conference and for this very informative discussion. Thank you very much.
Thank you.
Thank you.
Robert.